It's my pleasure to introduce our next speaker, Dr. Michael Gore. Dr. Gore is an associate professor of molecular breeding and genetics for nutritional quality at Cornell University, where he's a member of the faculty in the Department of Plant Breeding and Genetics. He got his bachelor's and master's from Virginia Tech and a PhD from Cornell University. So with that, I'll turn it over to you, Dr. Gore. Let's welcome Dr. Gore, please.
It's wonderful to be back in Iowa. So the last time I was in Iowa was in 2003 when I worked for Pioneer Hybrid. And after that, I went to Cornell University to pursue a PhD. And even though I only lived in Iowa for 2 years, it definitely felt like home. So I almost feel like I'm coming home. So I want to thank you very much for the invitation to give a talk on my research today. And I'll hope you'll have lots of questions for me as my talk goes on. So the title of my presentation today is Closing the Gap Between Genotype and Phenotype in an Era of High-Throughput Plant Biology. So we have heard a lot of talks about high-throughput phenotyping, so I guess I will add my flavor as well. So where are we going with this presentation today? We're going to begin by talking about nutritional quality traits in maize grain, and that would be for vitamin E. And then we are actually going to change it up and talk about cotton. So there's probably not a lot of cotton grown in Iowa unless you go to maybe a greenhouse on campus. But so my first job in Arizona after my PhD was to work on heat tolerance and drought tolerance in cotton. And that's where I began to work on the high-throughput phenotyping questions that I'll share with you today. So part one, maize. We all love maize where we're here in Iowa. So the maize that I love is orange maize because in our research program we've been breeding for high provitamin A. So maize is a global crop. It accounts for 15 to 56% of the total daily calories of some African and Latin American countries. However, in these countries, maize varieties typically do not provide grain with adequate daily levels of essential nutrients. There is tremendous genetic diversity in maize, as we all know. If you compare two maize inbred lines, there is divergence between a human and a chimpanzee, as Ed would say, my former PhD advisor. So how do we harness this tremendous diversity in the maize genome to develop maize grain that is enriched in nutritional quality? One possible way, and as I pose this in terms of the biofortification approaches, this is one of possible approaches that can be used to combat nutrition in the world. In a sense, there's obviously multiple ways that we can address nutritional problems in the world, be it through pills, food that is enriched in their nutritional quality, say through the processing of it, or through plant breeding. And that's where my research comes into play. So we can ask the question is, what are some of the major controlling genes for important nutritional traits in our crops? And then we can then begin to breed for this by increasing their nutritional value in certain varieties, say, that are growing presently in sub-Saharan Africa. So after we find these target genes, what we then want to do is find the most favorable, if you will, extreme alleles in the germplasm, and then bring that together with the germplasm that is being grown in, say, a country like Kenya. This has the potential to be cost-effective, and you can think of a seed as being a sustainable technology, as if it's, say, a maize inbred line, it can be self-pollinated and increased year after year. So, as I said earlier, this is not a panacea. This is one of the possible tools that we can address the nutritional problems in the world. So some of our previous work on provitamin A carotenoids, I'll just briefly touch on in the maize work. So carotenoids, such as beta-carotene, exhibit provitamin A activity. So this is the common provitamin A carotenoid that most people talk of when they talk about nutritional value in terms of trying to enrich grain quality for provitamin A. Sadly, if a young child does not receive enough vitamin A, i.e. through maybe provitamin A in maize, there are tremendous repercussions and consequences in terms of their eyesight and their health, and increased mortality rate as well in human populations. So some of our work showed we identified 2 genes in the carotenoid pathway, and these 2 genes have been used by Harvest Plus to actually increase the amount of provitamin A content in the maize grain 4 to 5-fold. So this we have kind of used as our model going forward. Yes, this is a somewhat achievable goal that we can begin breeding for nutritional quality in maize grain with a focus on sub-Saharan Africa. So some of the new unpublished work which I would like to talk about is vitamin E, which are also known as tocochromonols. So these are antioxidants, and they consist of tocopherol and tocotrienols. The alpha-tocopherol has the greatest vitamin E activity. So when people talk about vitamin E, they're often referring to alpha-tocopherol. So vitamin E, as you know, is an essential nutrient, but its deficiency is not pervasive as like provitamin A throughout, like, say, sub-Saharan Africa and Asia. But there are certain segments of the human population where this has been a issue. And some of the consequences in terms of human health in populations are increased cardiovascular disease and decreased immune function if there isn't enough vitamin E in the foods that they take in. So when we think about the extent of phenotypic variation for maize, and you can think of maize as being temperate and tropical and having a tremendous footprint in where it's grown in the world. So our first question is, what is the extent of alpha-tocopherol phenotypic variation in a maize germplasm panel that may capture about 80% of the common variation in the maize germplasm pool from like public, temperate, and tropical breeding programs around the world? So after we ran our HPLCs, and this is calculated BLUPs from 2 reps of the maize panel here grown in West Lafayette, Indiana, i.e. Purdue University, we found an average of 8.19. But what does that really mean in the context of human health and the food that one eats? So if we have a biofortification target of 38 micrograms per gram dry weight, and you're eating 400 grams per day of maize, so that's on average approximately a 5-fold increase. So if you look at a 38 microgram per gram concentration, our highest line here is at 30, okay? So in the present germplasm, so in the inbred lines that we're actually testing, we don't even have the proper combination of alleles to exceed our present target combination. So then the question that we asked ourselves is, first of all, we have to find those target genes, and then we have to mine those alleles to put them in the most favorable combination so that we can reach our breeding target for human health. So our favorite process or population at Cornell is the maize NAM population. And since coming from Cornell, I'm required by law to always show this slide. That isn't true, but I sometimes kind of feel like it. Anyway, so here we have 25 diverse founder lines, which capture maybe about 80% of the common variation in the maize germplasm pool. And they were crossed to B73 as a common parent. And each of these families consists of 200 recombinant inbred lines for a total of 5,000 recombinant inbred lines. So you can kind of think of the maize population as a giant sledgehammer for any complex trait that you can even think of, or possible complex traits that we haven't even conceived and won't conceive for like the next 10 years. So this is a tremendous community resource for the world. And this has been used to analyze such, I'll say, traits that can be more easily phenotyped in the field with a human eye, such as flowering time, leaf architecture, plant height, kernel composition, and leaf blight. So now we've grown 2 reps of the maize NAM population. This was our colleague Torbert at Purdue University. So now we have 10,000 grain samples, and we need to run HPLC on this. And that's what we have been carrying out for the past 2 years with like 3 or 4 HPLCs and a very hard-working group, to say the least, because having to run, actually with checks and controls, it's actually closer to 20,000 HPLC runs. And then after you take all the runs, then you have to occasionally manually do the integrations of all the peaks. And this is especially challenging for all the carotenoids because maize has tremendous variation for those compounds. So in the red here we have tocopherols, which are mainly in the embryo of the seed, and tocotrienols, which is blue, we're calling T3, mainly in the endosperm of the seed. And what you quickly gather from this pie chart here is gamma tocopherol consists of the vast majority of tocochromonols in maize grain. So that's about half. So alpha tocopherol, as we said, has the highest vitamin E activity here. So there's nearly a 5-fold difference between the content of gamma and alpha tocopherol content. And then when we look at the vitamin E activity, it's almost 5-fold as well, whereas alpha tocopherol has 100% vitamin E activity, and gamma tocopherol here maybe has only 8 to 19% vitamin E activity. So as you see, when you think about the breeding questions, what we really want to do is, if you will, change the composition of the maize grain, but in a sense of driving it towards increasing amount of alpha tocopherol. So how do we do this in the maize NAM population? First, we map QTL jointly across the maize NAM, and this is a model using 14,000 SNPs. So one key point here is this is using all of the recent recombination that was generated through creating this resource. And then we follow up with the GWAS. So this is now using the ancient recombination that has taken place in the population history of the maize founder lines. So here, since we're trying to harness all of that, if you will, ancient recombination, the rate of LD is going to decay faster. So here we're hoping to have enough mapping resolutions to find the genes underlying the QTL that have been detected. And being able to do this is also in incorporating our results from the joint QTL mapping in NAM. So here's kind of where the sledgehammer approach comes into play. We're using 28, not 1,000, but 28 million SNPs to do the GWAS across the 5,000 recombinant inbred lines. So when we're at first just doing the mapping with the 14,000 GBS markers, and in a sense we just want to see where the QTLs are in the genome. So this is just using the recent recombination here. And here in this cartoon we have B73, which is what I refer to as a black and white graph right here. And each of the 25 diverse parents here are P1 through P25. And using this B73 rare SNPs, we're able to track the chromosomal contribution of each of the 25 parents and the common parents, because each of these SNPs is rare in the sense it's polymorphic between B73 and all of the other 25 parents. Okay? So what that actually allows us to do is then being able to know what parent donated a particular chromosomal region. But since we are using the recent recombination here, we're going to have these large recombination blocks. So in a sense, most populations, when you QTL map, say, with 200 recombinant embryo lines, the level of resolution that you may gain could be half of a maize chromosome. So there could be like 10 to 200 genes in a particular QTL region, depending on the recombination landscape in that particular chromosomal segment. So now, for the 6 tocochromanol compounds that we analyzed by HPLC across the 2 reps of the maize NAM population that were grown in West Lafayette, Purdue, we detected a total of 100 QTL. And these traits were actually highly heritable, ranging from 0.71 to 0.88, and that's heritability in the broad sense. So if you look at alpha-tocopherol here and the total number of QTL that were detected, we detected a total of 13 QTL. And as we walk through this pathway, I'll kind of have the comparison between alpha-tocopherol and gamma-tocopherol, since that's, if you will, the shift that we're trying to breed for in the maize grain. So, sorry. And we found a total of 21 QTL for gamma-tocopherol. So when we looked at the effect estimates coming from all of the 25 diverse parents, we quickly saw that for alpha-tocopherol, which is on top here, and gamma-tocopherol, which is on the bottom here, the vast majority of the QTL effect sizes are very small, and then you see a tail of larger effect sizes. And that tends to predominate most of the complex trait variation that we have seen for traits that have been analyzed in the maize NAM population. So there is no one massive effect QTL that you can just use for this particular complex trait. Now, this trait may not be as complex such as a trait by flowering time, but there is a range of 13 to 21 QTL just for these 2 particular compounds. So we now wanted to ask the questions. Okay, we know the contributions of the QTL from each of the 25 diverse parents here. So then we asked the question, for the alpha-tocopherol content in each of these 25 lines, what is the QTL composition? So since one key point here is everything is compared to a common reference parent, okay? Our first example here is NC350, and in NC350 here, we have 3 QTL, which have a positive effect, and 1 QTL that decreases the amount of alpha-tocopherol content in maize grain. But obviously, all QTL effects aren't equal, and in contrast here, we have CML247, which has 3 positive effect QTL, just like NC350, but as you sum the positive effects of those alleles, they do not sum greater as NC350. So obviously, all QTL effects are not created equal. And here we have 4 QTL in this line, CML247, which are decreasing the amount of alpha-tocopherol content. So in terms of breeding, when you're thinking about recombining favorable alleles, then you need to think about this very carefully, because there are obviously a lot of favorable alleles, and all of this is in reference to B73. So when you begin to move these alleles around, you're moving them into new genetic backgrounds, such as lines that are now grown in sub-Saharan Africa. So even though we see a particular effect estimate in this germplasm, I think it's still an unanswered question of what effect estimates and what type of gene action we're going to see when we transfer it to new germplasm. So for these particular QTL that we're finding for alpha-tocopherol content, I am almost positive that there's going to be other rare QTL out there in the germplasm that we have not found yet. So even though, such as like this one line here that has one QTL and the additive effect may be small, we may find certain rare alleles out there in the maize germplasm pool that could have a very large effect just for the one QTL. So that is, I think, one of the important questions when we think about plant breeding is what is the contribution of rare variants to complex trait variation? And I think this is also very important and germane to nutritional traits as well. So now, with having the maize NAM populations, you can think of having 25 degrees of freedom, and we're able to test for pleiotropy in the pathway. So I would first like to draw your attention to gamma-tocopherol, gamma-tocopherol, alpha-tocopherol, and alpha-tocopherol. So what we're able to do here is just test for genetic overlap between the traits through the correlation of the QTL effects themselves. So this gives us a powerful genetic system to actually test for pleiotropy. But here we're actually testing for pleiotropy with the QTL effects themselves. So with the focus still on alpha-tocopherol and gamma-tocopherol, we can see the phenotypic correlation across NAM in red here. That's R. It's actually rather very low, 0.12. And the percentage of shared QTL, in a sense, pleiotropic QTL between these two traits is only 22%. In contrast, here we have alpha-tocopherol and gamma-tocopherol, and each of these, as I was saying earlier, the alpha-tocopherol we found 13 QTL, gamma-tocopherol we found 21 QTL, and the NAM phenotypic correlation is actually 0.07. So here, the actual percentage of shared QTL between these two traits is only 30%. So now that we have found all these QTLs, one of the first questions that we always ask is, what are the underlying genes for these QTLs? Okay? Because in this particular approach, we want to actually drill down to the gene, characterize those alleles, and then, put them in the most favorable combinations in germplasm, say, for sub-Saharan Africa. So when we began to look at the intersection of, if you will, the physical proximity of these QTLs in the maize genome, and then compared their positions to the positions of known candidate genes from precursor pathways and the tocochromanol pathway. Here are the precursor pathways. We have the MEP pathway, the aromatic amino acid, and the GDP phenol pathway right here. And all of the core tocopherol and tocotrienol enzymes are in black here, and these are highlighted right here. Something that quickly we realized, okay, so of those 100 QTL, 13 QTL map to the same region as these core enzymes here, which makes perfect sense. And then the other 20 mapped in this blue and purple region right here. But the vast majority of our QTL did not co-localize with any of the known candidate genes from these very well-characterized pathway. So 2/3. So 2/3 of the QTL are not associated with any of these biosynthetic candidate genes. So now we want to begin to actually drill down and to see what are the underlying genes for these particular QTLs, and that's where we begin to use the HapMap V1 and V2 SNPs, and then do the projection from the parents onto their progeny of 5,000 recombinant inbred lines. And we're able to do that through whole genome resequencing of each of the parents, And since we already know where the vast majority of the recombination breakpoints are from genotyping with 14,000 GBS markers, we're in a sense able to take those haplotype blocks from whole genome sequencing and then just do the projection so to keep increasing our resolution. So here, our previous resolution is, if you will, from what most people do with a recombinant inbred line population of 200 rils. So now we're beginning to use the NAM resolution, which is like the ancient recombination from over the actual population history. So the first example here is showing GWAS for alpha-tocopherol content in maize grain. And I'll walk you through all these axes here since we have a left and right Y and an X axis. So just to kind of ground you, the X-axis here is the 10 chromosomes of maize 1 through 10. And over on the right-hand side is the minus log p-value from QTL mapping jointly across the families. On the left-hand side here, this is a resampling probability, which ranges from 0 to 1 for our GWAS. In a sense, it's actually counting like out of 100 times how many times our particular SNP marker is in our GWAS model. So if our particular SNP is in the GWAS model perfectly 100 times, then that has a value of 1 here. So what I would first like to draw your attention to is this massive hit on chromosome 5. So this particular QTL in joint mapping across the 25 NAM families explains 80% of the phenotypic variation that we have seen for alpha-tocopherol content in maize NAM. So in a sense, we have been able to find this massive QTL, okay? But we now want to understand what is the underlying gene. So here we begin to use the GWAS approach, and our top SNP is within ZMVTE4. And ZMVTE4 is crucial in terms of the tocochromanol pathway because it is the key enzyme that converts gamma-tocopherol to alpha-tocopherol. Okay, so when you think of a breeding program and you want to design like a pathway breeding approach for your particular nutritional compound, you want to have a massive impact and you want to be able to, in a sense, shift the flux of your metabolite pool from gamma-tocopherol and alpha-tocopherol. The best way to do that is getting extreme alleles of ZMVTE4 and increasing the amount of alpha-tocopherol content here. So here we have gone from like running close to 20,000 HPLCs, doing joint mapping across the maize genome, and then the GWAS being able to characterize what the causative gene is. And then we've also have characterized this gene further in the panel of 252 lines, and we have an idea within the gene of where the causative variant is. But this particular gene is actually rather challenging because there's at least 3 or more causative variants. And a few of them are likely rare variants. So I think as we begin to characterize more QTLs in maize, this may be a very common outcome. So now we wanted to ask the question is this particular QTL expression-based? And to be able to do that, what we are doing now is correlating the peak QTL, chromosome 5 QTL effect estimates with gene expression levels. And what we've done here is sampled grain samples on certain days after pollination, 12, 16, 20, 24, 30, and 36 from all of the 25 maize NAM founder lines. So right now when I'm asking this question, I'm just looking in all of the particular QTLs in the support region surrounding the QTL, okay? And then I ranked it under particular R value. And our top hit turned out to be the ZMVTE4 transcript. So what we can conclude from this is this actually appears to be an EQTL. And then even though it's 25 lines, I then also use the FPKM values as a phenotype just to see where the peak is in the ZMVTE4 region. And it turns out the peak hit for the FPKM values turns out to be in the cis region of the ZMVTE4 gene. And since it's red, that means it's positive, okay? So the positive effect QTLs correlate with the higher FPKM values of ZMVTE4. And just one more example here for gamma-tocopherol. The top QTL for joint family mapping has also a very high PVE of 51%. And then when we did a GWAS, our top SNP was less than 100 kb downstream of the gene ZM-HGGT1. And this particular enzyme is very critical because if you look at ZM-HGGT here, this enzyme is particularly important for going into and making the precursors for tocotrienols right here, and then gamma-tocopherol right here. So here we now have a GWAS hit, and it makes perfect sense, okay? And then we ask the same EQTL questions, and it turns out ZM-HGGT1 is our top hit. So this also appears to be an EQTL, and it's positive. So that means like the positive effect QTL alleles correlate with higher FPKM values for ZM-HGGT1. So some of the conclusions we can draw from this metabolite QTL work in the maize NAM population is here we found a range of 31 to 21 QTL for the 6 compounds. So if you were to compare it to a trait such as like plant height or flowering time, these traits are not highly polygenic. And there's an emerging trend that expression differences may be the basis of most of the QTLs, and that trend seems to be holding up for genes that are in the actual pathway. Since 2/3 of the QTL did not map to any of the known candidate genes, there is still a lot we don't know about in terms of the control of this pathway in maize grain. So some of the future directions, we are also beginning to look at Q by E interactions, two-way epistasis as well. And since we have 2/3 of these unknowns, we're beginning to use co-expression programs and software to try to identify what some of those causative genes are under those QTL peaks, since we have no real candidates. So we're trying to use the actual expression levels of those particular genes in that region as one added phenotype to try to resolve that QTL down to the causative gene. And in the future, but obviously this would take like 5 to 10 years of time, and if you want to clone some of these QTLs and characterize the alleles in E. coli to understand what some of the causal genes are and the variants themselves. And so the key and what's really important is, okay, so we can find all the QTLs controlling these important new nutritional compounds and come up with a pathway breeding approach, but really it's then finding like the most positive alleles and then seeing how they perform in like sub-Saharan African germplasm. And that's some of the research that we're following up now with Harvest Plus. Okay, halftime. So I'm now going to begin talking about some of my cotton work when I was working for the USDA down in Arizona. When I think about the rationale for doing my research, I think of the typical cotton farmer that has to endure like a climatic drought maybe every 5 to 10 years, with their cotton fields. And when I was hired, I was hired to work on drought and heat tolerance to cotton. And I quickly realized that to have an impact on such complex traits, to improve the productivity of a particular crop, which farmers rely on and their families rely on, we're going to have to come out with new cutting-edge approaches. And this is one particular slide that Aaron showed earlier. And when thinking about maybe using a GS approach and coming up with a faster approach to develop crops that are tolerant to, like, if you will, climate change, heat and drought tolerance, I began to think about, okay, so there's certain areas that need some attention in these GS models. And I'm not going to dwell on this too much, but of the two that I think are very critical is when you think about recombination, and that is the evolutionary engine of all plant breeding populations. So you want to accelerate the recombinational process in your breeding programs. And in purple over here is in particular the area that I've been focused on, and that's the phenotyping. So being able to improve phenotyping capacity and high-throughput phenotyping. So 2009, I'm out in a cotton field, really hot, like 115, doing some manual phenotyping of cotton plants. And I quickly realized I'm not very heat or drought tolerant in Arizona. And if I'm going to survive out here in this very harsh climate that no human being should actually be in, by the way, I'm going to have to come up with an intelligent approach to do phenotyping, or I'm just not going to last out here. So I looked across the field, and what did I see? A tractor owned by Pedro Andrade Sanchez. He had this crop circle instrument mounted on his tractor driving through the field. And that was kind of like my eureka moment and almost my life preserver at the same time. So I ran over to Pedro and Andrade and was like, Pedro, we really need to collaborate. I'm like, I do breeding and all this. I realize you're an ag engineer. You don't know anything about breeding at all, but let's sit down and have a conversation. And that conversation turned out really well. And one of the first, this is like version 2 tractor. So our version 1 tractor was actually a tractor we pulled out of the trash. And we had to put in a new engine and all this equipment, got all grease all over ourselves. It was all fun. But that is how all of this kind of got created. So some of the first traits that we were targeting were canopy temperature with infrared thermometers, ultrasonic transducers for plant height, and these crop circles for vegetation indices, which were covered very well earlier by Aaron. So for our high clearance tractor here, we're able to do 4 rows at a time. And here we have the GPS RTK antenna and GPS RTK receiver and radio. And we were able to collect at 1 hertz driving through the field. And just to kind of give you a frame of reference here, this is a shroud, if you will, kind of covering the ultrasonic transducers. These are the IRTs. One's pointed down at the ground, one's at a 30-degree angle pointed at the vegetation. And these are the crop circles here. And then we were using CR100 Campbell Scientific Data Loggers to record everything, and we also had laptops as well. And this just came out 2014, this year, in Functional Plant Biology with Pedro Andrade as the first author. So why is Central Arizona such a good place to do high-throughput phenotyping? Clear skies, not very much rainfall, and high temperatures. We do, in fact, have the monsoon rains towards the end of July and August, but in general, the soil is very sandy, so when it will actually rain, it just drains out very fast. And we had 2 treatments here for a recombinant inbred line population of only close to 100 rills. So this was a very humbling time for me after coming from Cornell, where we had access to 5,000 recombinant inbred lines. Here I was using 100 recombinant inbred lines, but at the same time, it was the perfect size to ask the questions that I had to at that time with this particular platform. So we had two treatments here, wet and dry, and if you will, recreated a heat map of canopy temperature of the field. And here's a rep 1, range of 31 to 46 degrees C, and this is the dry plots. So I was really excited when there was talk about the EM38 earlier, because I was trying to figure out what was going on in this part of the field. But when we took an EM38 through the field and looked at the conductivity of the soil, I found out that there was a higher clay content in this patch in the field. So if you're doing any droughts work in your field, I strongly encourage you to characterize your soil profile, because there's more variation for your soil than you will have in your plant population. And here is rep 1, and this is blue, and this is our wet treatment here. And this is rep 2, and this is our dry treatment. So if it's green like this, it's hot, and here's rep 2, wet. So the first question we wanted to see and ask is this particular high-throughput phenotyping platform with the IRTs able to see a change in canopy temperature over time? So out in Arizona, breeders have been unconsciously selecting for certain genotypes of cotton that will transpire at a high rate, because any crop that's grown out in like the region outside of Phoenix where we are, and for the vast majority of Arizona, is all under flood. Okay, so breeders, since like the 20s, have been breeding for plants that'll transpire at a very high rate, so that they're able to cool the canopy. Well, what happens when you begin to test these genotypes under drought? The transpiration rate is decreasing because the plants are shutting down. And that's, in fact, why the canopy temperature is increasing. And here we have the greatest increase from 10 a.m. to 1 p.m. And this blue is the wet control. And this black line is the ambient air temperature up here. And there is a time by treatment interaction. And so we have this recombinant inbred line population, so then we can ask questions about how heritable this variation is. And under the wet treatment, so these are coming, so this was on 2/24 at 1 p.m., wet in blue, dry in red. And so these are the actual values of the recombinant inbred lines average across the two reps. And here we have an average of 32 degrees Celsius for the wet plots. And you can see the transgressive variation. These are the two parents here, TM1, NM2416. And here are the dry plots with the TM1, NM2416, and the parents are keeping the same rank. And that had a mean of 38 degrees Celsius. And that's at 1 p.m. And it was actually heritable. So that was like the big relief, if you will, after putting in all this time to get this tractor out to the field and have all of this equipment, to actually see it had a broad sense heritability that was at least somewhat okay, of like 0.52 to 0.58. That at least gave me some confidence to move forward then. And then I also wanted to ask the question how repeatable this is from week to week. This is day 217, and what we have here is the ratio of 1 p.m. dry over 1 p.m. wet, and that's canopy temperature. And here we have 1 week later for the same trait, and we have an R square of 0.47. So how I kind of view this is the phenotypic consequences of this heat and drought treatment are pretty much the same from week to week. There isn't much change when you're in a place like Arizona. It hardly ever rains, it's always hot, and the sun's always out. And now I wanted to begin doing time-related QTL mapping, because if we have this tractor platform, we can actually see which QTLs are turned on at certain time points on a particular week. So just to kind of walk you through this, this is QTL mapping, and we had genotyped this particular cotton recombinant inbred line population with Jesse Poland So he was also brought up in some of the prior talks. So this cotton population, so it was 100 recombinant inbred lines. So let's just focus on this one particular QTL here. So if it's red, that means it's a positive coming from line TM1, and that means the canopy temperature will increase. And this particular QTL, its strongest expression is at 1 p.m. in the afternoon on week 217. And then the following week, day 224 at 1 p.m., the same QTL is showing up. And then this QTL on chromosome 10 is also showing up and showing the same pattern. But then we also see cases where a particular QTL is just showing up at one time point for one particular week. So there is variation in which QTL are passing our 5% FDR. So Aaron also had covered some vegetation indices. So I had all this NDVI from the crop circle. And then I kind of wanted to ask the question, what else could I do with this than just do mass, if you will? So I calculated an index that can, in a sense, quantify the degree to which the plants wilt. So how this works is we take the NDVI value in the PM for the wet plots, subtract it from the NDVI values PM for the dry plots, over the NDVI PM values for the wet plots. So what's happening here is as the plants are undergoing stress, the leaves wilt and become very droopy. So as the tractor is driving over with the crop circle, what happens when you're looking down over top of the canopy is you're seeing more soil exposed, right? So if this particular index is, in a sense, quantifying the amount of greenness, then you would hypothesize that amount of greenness is going to decrease as the plants wilt. And that's precisely what we saw here. So if you look at the red line here at 9 a.m., this is for the drought plots, you can see a dramatic increase in the NDVI value from like 0.72 to 0.68 almost at 1 p.m. And the plots that are wet, there's a slight increase in their actual NDVI value. And what I would maybe attribute that to is what happens is the plants are like tracking the sun throughout the actual day. So then they're changing their like geometry and their orientation of the leaves. And this particular trait was actually had a higher repeatability than canopy temperature as well, with the R square of 0.76. And then we also mapped QTL in a time-related QTL approach. One QTL I would like to particularly focus on is this one on chromosome 10 that I have marked here in red, because this QTL was also detected for canopy temp as well. So it's possibly a case of pleiotropy. The only other QTL I would like to highlight is this one on chromosome 25 that tends to be showing up on these 4 time points here. On particular day 224 at 1 p.m., this intensity of the drought stress was the most intense of all these time points. And we see that this QTL isn't really clearing the FDR here, but then these 2 QTLs are showing up at this time point. So there's very much, if you will, some G by E, and the only way we're able to really quantify that is with this tractor platform driving through the field. Because if you think about a trait such as canopy temperature, trying to go out and actually phenotype canopy temperature with handhelds over like a certain block of time, and if you have like a field trial of thousands of plots, you would almost need like an army of people. But this provides an opportunity to like fastly go through the field and get highly heritable phenotypic information. So some of the next steps for high-throughput phenotyping in cotton is now to relate a lot of these phenotypic parameters to yield, and that's being carried out by my colleague Andy French in Arizona, and that first yield trial was last year, and there will also be one this year in 2014. And working with Ivan Baxter at the Donald Danforth, we're beginning to look at the ionomic profiles of the seed. So if you have magnesium or iron or zinc in the field, in the seed, I mean, we're asking the question, is that something that can be used to track how the plant is responding to drought? And is there a certain profile that more drought-tolerant cotton plants actually have? And with Andrew Patterson at University of Georgia, we've been creating very large cotton populations. So we plan on growing these out in Arizona and probably also Texas and Georgia in 2015. So Cotton Incorporated is beginning to invest heavily in some of these high-throughput phenotyping vehicles, so we can begin to address these same questions in a very large cotton dam population. So some of what I've also been playing with is looking at other phenotyping technologies that can be used for high-throughput phenotyping in cotton, but also high-throughput phenotyping in other plants. And the LiDAR, Aaron, as you were showing earlier on the nice drone, here we had actually put it on our tractor. And here we're asking the question, can we do canopy geometry? And this was with my colleagues down in Arizona, pretty much led by Andy French and his programmer. Here we're looking at canopy height and canopy width. And as this particular LiDAR instrument is mounted on a tractor and it's collecting phenotypic data so fast, just after a few passes, we can have a terabyte of cloud points. And so it's been quite a computationally challenging venture to try to sift through all this information and get something that's actually biologically value out of it. So what we're beginning to use these cloud points for is now to recreate the canopy geometry of these cotton plants from the tractor as we're driving through the field. And right now what I'm showing you is actually kind of crude, but what we're actually hoping for in the future is then to do canopy height and canopy width, and then over time doing branch angle, and in the future possibly the size of the leaves. And the ground penetrating radar, which was talked about earlier, my colleague Sean Thompson has been using the ground penetrating radar to do root phenotyping. And I don't think this is quite prime time yet, but he has made a lot of great progress on it. So when you think about able to do root phenotyping, such as with ground penetrating radar, then you can begin to look at the various horizons of the soil and then look at the mass of the roots at the various horizons. And that's, in a sense, what you're kind of seeing here in these heat maps. So as I said, don't be really quickly just to jump out and go purchase a ground penetrating radar right now to do root phenotyping. It's at a stage where it's a possible tool that can be used, but there's definitely some challenging computational work that needs to be carried out first before it can be doing very finely resolved root phenotyping. And of course, the hexacopter platform, this is with Kevin Price at Kansas State University. So I've been in close contact with him, and this is a platform I would like to begin using at Cornell. The tractor platform may be a little more challenging at Cornell because we have hills, those rocks, and I also want to be trying to use this on maize. So you can think of something very tall at an angle probably would not be very safe. So that's one of the reasons why we're beginning to look at this drone technology for phenotyping at Cornell. And tractors also have the challenges of compaction, plant breakage, and when you're doing drought work, you can maybe only go into the field when the field's actually dry, or you will certainly tear your field up with a tractor driving through. And so when you think about being able to phenotype a particular plant over the entire growing season, then you can begin to ask some really incredible G/E questions, and where you can think being able to understand G/E in the sense of growth rate from the time of planting, and then how a plant dries down, and then at harvest, being able to understand, what is the phenology of this plant. And then some of the questions I'm very intrigued on is the quantitative control in terms of genes for the variation for these traits as well. And since I'm also working on nutritional quality, there's some now seed approaches out there that can be used to do geometry in a sense of size and shape, and also seed quality traits. And being able to then actually sort grain on their quality characteristics. So when you think of a GS breeding program, and then you can then quickly just scan thousands and thousands of seeds so that you can select a certain type of quality, and then you can begin to then use those lines to create new training populations. So I would like to acknowledge the tremendous support for PGRP and USD-ARS. My colleagues at MSU did the vast majority of the HPLC phenotyping. Robin Buell did a lot of the computational work on the maize project. Torbert Rocheford grew the maize NAM population for 2 years at Purdue, and Ed has genotyped just about every maize plant in the whole world. Close to it. And of my colleagues in Arizona, I would like to say thank you to Pedro Andrade Sanchez for taking a risk and being able to work very closely with a plant breeder type, and being able to create this fantastic high-throughput phenotyping platform with me. And my colleagues Andy French and Jeff White have been very supportive over time, and in particular Jesse Poland for helping with the GBS of the cotton recombinant inbred line population. Thank you very much for your time.
Questions for Dr. Gore?
Hi, Mike. Awesome research. I wanted to ask a little bit about your nutritional quality work in corn. So, for example, the fact that you were able to hit, what was it, VTE4,
Yep.
right, right on the head.
Yeah.
I mean, it really speaks to the power of genetic dissection that you've got with the NAM and with that genotype data. But from a phenotyping perspective, I think the real elephant in the room associated with phenotyping for nutritional quality traits is bioavailability. Right?
Yeah, that's right.
Vitamin E and vitamin A are both fat soluble, but the population you're targeting doesn't have a very high dietary fat intake.
Yeah, so if they're getting enough fat in their food, then it's okay. So I guess one approach I've kind of talked with Torbert on this is maybe breeding for high oil maize as well, in parallel with high provitamin A maize. But yeah, it is definitely an issue when you have populations that subsist almost entirely on maize grain. Being able to bring in other foods, it's pretty challenging. Yeah.
And do you think, is there a correlation or even an association? I mean, at some level, I'd like to believe that if you're increasing content, maybe the bioavailability is expressed as a % of content, right? So increasing content does at some level increase bioavailability, but there's got to be a point there where it just passes through the diet if it's just not soluble.
Yeah, I haven't seen any work on that actually in terms of seeing how far you can push the amount of like provitamin A and then seeing what's actually retained. I myself haven't seen anything on that. So what we talked about yesterday was, since there's so much like turnover of carotenoids themselves post-harvest, that breeding for like higher total carotenoids and higher provitamin A is very critical. In a sense, we have like a harvest plus breeding target now, but being able to breed beyond that to account for that actual loss after harvest, after the grain's cooked and processed, because there's an enzymatic process that breaks down the carotenoids, and then it's coming from like heat and light, and then there's just like a first order reaction that's non-enzymatic as well, that's breaking down carotenoids. So I think that is maybe more of a greater challenge right now that's pressing before the bioavailability part, but that part is actually maybe more critical for like zinc and iron right now. Yeah.
From a logistical point of view, when you're doing your monitoring in cotton, if your prime time is 1 o'clock,
Yep.
do you do a time sequence analysis so that if you start at 12 and end at 2, you do a regression, or how do you do that? Because you can obviously not do everything exactly at 1 o'clock.
Yes, so we have a like time term in the model. So we have like GPS coordinates, but it's also time stamped, and so we're able to use that as a cofactor in our models.
So if your ideal is 1 o'clock, about
Yep.
what is your prime time? Is it a wide window for a couple hours on either side, 1 hour on either side or so?
Yeah, that's a really good question. So we have done at 7 a.m., 10 a.m., 1 p.m., and at 3 p.m. From what I've seen, kind of the prime time is after 10 till close to 3. And then after that, things begin to kind of cool off some. And then in the evenings, these plants are so resilient in the sense they can rehydrate at night and they look fine early in the morning. So it's really that like peak afternoon when you see like the most intense like consequences from the heat and drought treatment. Yeah.
There are no other questions from the audience. We have a couple here from cyberspace. The first is the version 2 phenotyping tractor. How much did the implement itself end up costing with the camera and software equipment and without, and was the frame custom built?
The frame was custom built. All of the equipment, so I actually have like an Excel sheet because I've been sharing that with folks on like the cost. So if, say you can get like a used high clearance tractor for maybe 20, 25 grand or even lower, all of the other equipment, depending on like the quotes that someone can get, you can probably recreate our system for less than 80,000. And I think it'll get cheaper in the future. So my colleague Jeff White is actually testing new equipment that's cheaper to even drive the cost down lower. Yep. Yes, Jonathan.
Mike, thank you for your presentation. The last question prompted me to think, obviously you're trying to track processes that are very time sensitive in the middle of the day. I've done those kind of measurements by hand, and every 10 minutes things are changing. But then if you go into pre-dawn measurements, the plant's at a nice stable, from 2 to 5 a.m. Now, you're not measuring the same things, but if you can get some measurement of, zap the leaf and look at water content. Have you thought about a pre-dawn type thing?
So like Jeff White and I have definitely have had that conversation, and what we would really like to do at some point in time is do the phenotyping every hour over like a 2 to 3 day period, pre and post drip. Oh yeah, so one key point here is all of this was grown on drip tape. So we had like precise control of all of the processes. So the key is I think there is definitely value from like 2 a.m. to like 5 a.m. I just couldn't find someone to drive the tractor at that point in time, to be perfectly honest. But I had actually thought of that. But yeah, I think with our version 3 tractor that we have now, I think it actually has headlights on it, so it would work. Yeah, but I think that's a great point, Jonathan. Yeah.
So there's one other question from cyberspace. You mentioned that part of the future plan is to apply this phenotyping platform to maize. How far along is your team on that effort, and what do you think will be the hardest part?
How far along are we? I recently posted for a postdoc to work on this project at Cornell, so if there's anyone that knows of someone intrigued of coming to Cornell and do some high-throughput phenotyping, then call me. So that's kind of where I am right now. And we are getting ready to hopefully purchase a GPS planter. So that's kind of like the first phase. And then I think as people drink our Kool-Aid and realize, oh, field-based phenotyping is where it's at, then I think a lot of people will begin jumping on our train. And once you have that like critical mass, then I think Cornell will be able to grow pretty fast. But right now, my first plan is to work closely with Kevin Price and use his drones that he has created for like wheat, and then try that at Cornell at maize this year. Yep. Okay. Jimmy?
Great presentation, Mike.
Thank you.
So what is your future plan that based on the time series QTL mapping analysis, where you show QTL showing up and gone, and then a robustness of this method? Because if you actually measure multiple time points at a day, and then you can have multiple days at different stage, that unless we actually upgrade our analysis framework, that now we have a correlated phenotypes with N and P together. If you apply FDR, it's going to kill all our signals.
There won't be any QTL that like pass then. Yeah, that's a really good point. Since I actually was having that problem, I guess the easiest way to get around that is treat each particular time point as, if you will, a trait of its own, right? But the challenge is I want to also bring in all of that information from the whole growing season, right? And QTL map with that. So that's actually a problem I have not been able to overcome yet. So if you have any ideas, please tell me.
What do you think about when we think things, we think differently than physiologists?
Yeah.
Like Kevin Price had this NVDR thing. How do they actually want us to integrate all this different time point measurement first before we apply our genetic analysis tools? What do they feel actually we're collecting all this data?
Yeah. I don't think I have, well, I guess this is kind of shooting from the hip, but I don't really have a concrete answer for you yet, Jimmy, because I'm kind of at the phase now is trying to figure out what the key time points are for a particular trait of a particular crop, right? I think if I'm able to address that question first, I won't bring in a lot of the other traits, if you will, from points across the growing season which aren't adding any value. And that's the thing that I'm seeing is when I begin analyzing everything from across the growing season, it doesn't make clear sense always, because each particular QTL is like turned on at like a certain point and time, right? But early on, these particular ones aren't turned on. Okay, so I think it's probably going to take more thought in how to partition that out across the whole season. And that part's not very clear to me yet. You could have some thoughts on that.
So that just relates to Jonathan's presentation.
Yeah.
If we actually treat each of the time point measurement as some fins, there's just so many fins that are underlying this composite trait that we're just mapping those fins individually.
Right.
Even the scanner, the equipment are the same, but we're measuring something different, like these other, colleagues ask the question.
Right.
Just a comment. Yeah.
Yeah. I mean, the quickest way is to do like a principal component, across the season. But I think there's more intelligent ways, and I myself, I'm just not there yet in what way that should actually be. Yeah.
Well, let's thank Dr. Gore for his time. So for those listening on cyberspace, we're going to take a break here for about 30 minutes. For those here, there's some light refreshments if you want to hit the restroom. We'll be back at about 3:30 for our final speaker.
Two places you can go. My passport's like auto hide, so it's not auto hiding right now. Alright, so if you want to use that, otherwise you can use a handheld microphone. So this will probably use that. You don't set up right now. Sure. It's live. But otherwise, so this is just a quick round like I've used it before. I went back, forward, back, it's embedded. That's choose which side to actually point at. Yeah. A lot of people have been doing this.
Yeah, I think whatever side you're going to be turning to, it might be, like, it seemed Dr. Gore put it, like, up on his cog, so if you're always going to turn to your right, that way then you'll lose the audio. Yeah. Well, it's because he had it on the opposite way he was turning, so if you're turning this way, it'll be easier, but if you're turning that way,
Yeah.
Yeah, it might work right there.
I'll talk loud.
I think either or. We've been able to hear everybody. So.